If you have your DNA Master program downloaded, you are ready to update it and set the preferences. So I'm going to begin by opening up my DNA Master program by clicking on the shortcut. And I'm going to slide this over so that I can see my DNA Master page and the instructions in the C-Phages Bioinformatics Guide. Where can you find the Bioinformatics Guide? That information, the website, is located in the syllabus, and it's also located on this assignment for today. If you go to this website here and you use this username, cphages@gmail.com, and the password aviator31, you will have access to this free guide. And I'm going to begin by clicking on Annotation Outline and going to Setting Up Your Computer. There's information here about installing DNA Master. And since I already have DNA Master installed, and you should too, we're going to go to Updating DNA Master. And this is really simple. We're going to go to the Help menu and select Update DNA Master. This should take a matter of seconds for it to update. We're going to say OK. And now I'm ready to start working on my preferences. So now I'm going to click on the instructions for DNA Master Preferences. I'm going to go to File, Preferences, and we are going to begin by clicking on the tab Local Settings. And we're going to click on New Features. And what we want to do is set the default translation table. We want to change it from standard code to a code that's consistent with bacteria and the viruses that infect bacteria. So we are going to select bacteria and plant plastid code. Make sure that Add New Features to Documentation and Add New Features to Features table is selected. Your default product for open reading frames of unknown function should say hypothetical protein. And finally, the instructions tell you to insert template text into your notes during the auto annotation. And this year, actually, we're going to do things different than what C-Phages recommends, because we're going to keep our notes in a different program called Pecan. So you can uncheck those two boxes. Next, click Apply. And now we're going to move on to setting the gene prediction server location. And this requires a document, which I also have attached to this assignment, but I'm going to demonstrate here. This is a document with instructions and passwords so that people in C-Phages have access to this particular database. So I'm going to bring up that document here. And now we're going to click on Preferences, and now we want to go to, let's see, we want to go to Internet, and we want to go to Gene Prediction. And what I'm going to do is make sure that the Glimmer server and the GeneMark server fields have the correct address, that they match what's here in each of these windows. These are the web addresses so that this program has access to these two web programs called Glimmer and GeneMark. And the other thing that we want to do is click on the box that says Use PBI Server for C-Phages course. Now this requires a password. So we're going to enter the code HATFUL in the field unlock code. And this is case sensitive, so make sure you use a capital H, A, T, F, U, and 2 L's. And now we are going to click on Apply. And so that should be all set. If we don't do this, your auto annotations won't work, so make sure you follow the directions here carefully. Next thing we're going to do is go to File, Preferences, Internet, and now we're going to click on Secure Connections. And I'm essentially going to uncheck the box labeled Secure Connection to the GeneMark server, which are this top one, and also the same for the Glimmer server. We're going to uncheck those. And I'm just going to leave these two boxes checked, Secure Connections to NCBI Genome Server and to QBLAST Server. And now I'm going to push Apply. All right, now I can get rid of this document and go back to the guide. Next, we're going to set the start codon choices. So we're going to go back to Local Settings and click on the Translation tab. And essentially we want all the boxes checked. This is telling DNA Master that we want to look for all 3 flavors of starts, ATG, GTG, and TTG, and all 3 flavor of stop codons, TAA, TGA, and TAG, TAG. Once this is all set and you have all these boxes checked, you can push apply. And now finally we're going to go to set where we want to save all of our DNA master files. So we're going to go to directories, which is this first tab, and I'm going to click on the archive button, and I'm going to browse to a place where I want to keep my files. And I find it easiest to keep them out of the DNA Master program. So the operating system of DNA Master on my Mac is within a program called Wine, and I'm going to navigate outside of that and keep them on my computer. I'm going to click on Users, Sally Malloy, Dropbox. And you guys can choose to keep them wherever you want to keep them. I'm going to keep them in, you could keep them on your desktop. I'm going to keep them in a folder where I keep all of the genome sequences. Right here. So I'm going to say OK. So finally, you're going to go to set default values for BLAST searches. I'm going to have you set the preferences for BLAST searches, even though it's likely you will not do it. So go to Preferences, Internet, and BLAST. I want to make sure that they're set as they are here. However, doing BLAST searches in DNA Master is really, really tricky in terms of it takes a very, very long time, and you often have to have these searches running overnight in order to get all the data back from NCBI that you need. Because this is so difficult, we will probably ask you to do auto annotations without using BLAST, and then provide you with the files that you actually need with the BLAST searches already, the BLAST data already included in the file. But just the same, to be on the safe side, in case you do choose to do some blasting from DNA Master, you might as well set this up as it suggests here. And then finally, you're going to go and set color preferences. And this is actually, it seems kind of minor having the colors that you like in DNA Master, but I actually find it very important because I sometimes cannot see things really well unless it's using the right color. So we are going to go back to local settings, and we're going to click on colors. Now I choose to have the colors for the ORFs, the reverse ORFs be red and the forward ORFs be green. But maybe you're red-green colorblind and you want to change those. You could change it to, I suggest, a very dark color. Otherwise you can't read the text that's associated with the ORFs. And I'm going to leave these as they are. They recommend different colors for these different CD frames, but I find that sort of less important. Once you have the colors the way you want them, you simply push apply. And once this is the end of setting up your preferences, and so the only thing you have left to do is push the OK button, and it says save changes, and obviously the answer to that is yes. So now you should be all set to open up files and do your auto annotations.